students, a warm welcome to the course Bioprocess Engineering. Today we are going to step into part 3 of Unit 5. Let us have a short recap as the topic that is being introduced to you today will be an advanced one which requires a sufficient recap. We have been studying in our earlier classes about the importance of mathematical modeling. Can anyone tell the basic needs for modeling approach? Modeling is carried out to improve process understanding of performance and predict the performance under conditions such as process design, optimization and control. And thirdly, in a mathematical model, we can have one or more equations that we can use to answer questions about the system by using minimum data compared to the empirical approach. Now, what is an empirical approach? Empirical approach is the theoretical method of predicting data in which the productivity is measured by combination of plant operating conditions and correlations were made. Next, modeling can be performed in various stages starting from proper definition of the problem, formulation in terms of mathematical terms and digital simulation using numerical method of solution and validating the solution depending on the correct choice of theory, the ability to identify model parameters correctly using numerical solutions, and finally care and judgment will be taken such that the model does not become over complex. There are two basic characteristics of any bioprocess model. First, a model can be structured or unstructured depending on whether it describes intracellular characteristics within the cell, and the intracellular characteristics in specific mean the metabolic reactions and cellular processes as occurring in the cell. Second, a model can be segregated or unsegregated model depending on whether it considers or not the heterogeneity of cellular population. Depending on whether it considers or not the heterogeneity of the cellular population, the position in the cell cycle, the choice among these properties depends on the objective of model, and such reaction rates vary with time and are usually influenced by many parameters such as substrate, biomass and product concentration, and also the physicochemical conditions such as pH, temperature, dissolved oxygen concentration and the concentration of various growth inhibitors. The models which incorporate the details of intracellular metabolism are referred to as structured models, and such models attempt to account for unbalanced growth of microorganisms, that is, when the composition of major cellular constants such as RNA, enzyme concentrations vary as a result of changing external conditions. The one such model that we have seen in the earlier class is the compartmental model, where these models attempts to include structure in models of cell growth and metabolism. The cell mass is subdivided into various components as S, K and G, where S is the substrate reservoir, K be the synthetic component and G be the genetic component. The stoichiometric balances in each of the compartment has a major impact in regulating cell's internal machinery and affecting the yield of any given process. Broadly, we have studied about 2 and 3 compartmental model. In 2 compartmental model, the energy flow between G and K compartment was studied briefly, whereas in 3 compartmental model, the mass flow from substrate to G and K and the interrelationships between the components was described. The major predictions of compartmental model is that it predicts the existence of initial lag phase. The cell mass will increase while the cell number will not change initially. During growth phase, both cell size and cell number increases exponentially, and the stationary phase is attained with respect to cell mass prior to cell number. Next, we have discussed about single cell models, in which the single cell models have the advantages that they can incorporate cell geometry and its influence on metabolic transport and they can predict the various temporal events during the cell cycle, and also they can incorporate details of spatial arrangements within the cell and includes the details of various metabolic pathways. The cell is treated as an expanding reactor and the mass balances are written, which include the effect of changing cell volume resulting in a dilution of intracellular concentration. The mass balance for DNA synthesis was written as a function of specific growth rate, concentration of deoxyribonucleotides, volume of the cell, and the concentration of glucose, and also the number of replication forks in the cell. These single cell models can provide information on the transient response of the cells to environmental changes and are capable of predicting measurable quantities such as cell size and nucleic acid content. Next, we discuss about plasmid replication model, where plasmids are extrachromosomal DNA developed in a bacterial cell in order to make the cells adapt to various stresses, and these plasmids will be replicating on its own and will be segregated into daughter cells based upon the law of segregation, and these plasmids plays a major role in production of recombinant proteins, and it is essential for any bioprocess engineer to know the rate at which the plasmids replicates so that the rate of production of foreign protein in a given host cell can be easily determined. But once these cells adapts to given environment, then the plasmid will be lost from the system, and the probability of forming a plasmid free cell is given by 2 power of 1 minus n. The plasmid replication depends on the initial number of plasmids and also the specific growth rate of these cells. The maximum specific growth rate and the saturation constants for plasmid replication and cell replication has been determined by using the linear equations, and we discussed about the plasmid stability model. As the cells adapts to the given environmental conditions, the cells have a tendency to lose the plasmids, and in certain cases the plasmids will not get properly segregated to the daughter cells, wherein the bioprocess engineers cannot easily predict the rate of production of recombinant proteins, as bioprocess engineers face two challenges: one with respect to rate of plasmid replication and next with respect to plasmid stability. Suitable models are developed in order to study the yield of a process in the presence and absence of plasmid. The key parameters of plasmid stability model is that probability of plasmid loss per generation of cells, relative growth rate of plasmid free and plasmid containing cells, and the rate of generation of plasmid free cells. Normally, a bioprocess engineer works with batch, fed batch and continuous system of cultivation. Batch system is the one where the substrate is added into the reactor and will be inoculated with the organism, and the optimum process conditions will be provided for the organisms to grow, and the process will be stopped once the cells attain its complete growth. In the batch system of cultivation, as the organisms grows, the substrate concentration dramatically decreases, whereas in the fed batch cultivation systems, we can observe the presence of lag phase, log phase, stationary phase and dead phase. In the fed batch system of cultivation, the limiting substrate concentration in the cell is about to complete, then fresh substrate will be fed into the system, wherein we are making the cells to prolong in the log phase rather than to switching on into stationary phase, whereas the biomass concentration increases as long as the substrate is available in the medium. In the continuous system of cultivation, the substrate is fed into the reactor and the biomass and the products will be continuously removed off from the system. In the perfusion systems, the cells will be fed back into the reactor wherein the product alone will be taken out. Concentrated feedback systems are carried out by providing a concentrated medium where the dilutions are strictly avoided. The major drawback with perfusion, fed batch and continuous system of cultivation is an increase in dilution of the system as the medium is fed into the reactor. The continuous stir tank reactors are used in most of the bioprocess in which the system will be fed continuously in circulation with the help of suitable agitators. But practically, it takes some time for the medium to get completely mixed up and we can observe the formation of dead zones, and these dead zones are the regions where there won't be any mixing. In the continuous stir tank reactor, which are commonly used in certain cases, bypassing may occur where the medium that is fed into the reactor will not go for process but rather goes out into the outlet zone by bypassing the motion. Modeling of bioprocess plays a major role in research and development. Optimize the bioprocess in order to maximize yield and also to scale up the given process and simulate the growth of organisms. Also, we are able to perform the online control and optimization process using soft sensors, and economic evaluation of any given process can be easily performed using modeling approach. Continuous stir tank reactors are preferred choice of reactors for high quality scientific studies for the production of various metabolites. The bioprocess are very sensitive to disturbances in environment. The continuous stir tank reactors can be predicted to have an increasing appeal in future bioprocessing of cheap raw materials to make high value products. It is consequently important, especially for the process control of these reactors, to analyze the dynamics of state continuous reactors. The process that has been modeled can be simulated using suitable softwares in order to estimate the optimum process conditions such as speed control system, gas supply system, temperature control, pH, dissolved oxygen, foam and various components of bioreactor, where here we are seeing that the flow rate of air can be more adjusted and we can estimate the process parameters. The temperature inside a reactor can be adjusted by giving proper hot water and cool water supply, and we can observe the circulation of water from the reservoir. The pH of the system will be adjusted according to the production of given metabolite, and if in certain case if the pH drops, you can see the addition of base into the reactor, and if the pH increases beyond 7, then acid will be subsequently fed into the reactor in order to maintain the optimum pH resulting in production of metabolites. Next, the dissolved oxygen concentration can also be measured and controlled, and the process parameters can be estimated for maximizing yield. So, the efficiency and the performance and efficiency of various parameters can be easily studied using simulation softwares. The entire system can be simulated by modifying the various physicochemical parameters, and the yield of any given process can be estimated. With this short recap, let us dive into the topic for day. In today's class, we will be studying in detail about the dynamic simulation of batch, fed batch, steady and transient culture metabolisms. In a batch system, the substrate will be fed into the reactor and reactor will be inoculated with the given organism to produce the metabolite, and based upon the product formation kinetics, the batch system of cultivation will be prolonged and the metabolites will be taken out once the process is completed. Till the process gets completed, there won't be any exposure of the system into the environment. In the continuous system of cultivation, the substrate will be fed continuously into the reactor and the product will be taken out continuously. The flow rate of the substrate and the flow rate at the outlet determines the dilution inside the reactor and the amount of substrate available for the cell mass to grow. There is a continuous exposure of system into the environment via the flow rate at the inlet and the outlet. In the feedback system of cultivation, as the substrate concentration gets exhausted, then the substrate will be freshly prepared and fed into the reactor. The addition of substrate in turn prolongs the log phase of the cells and makes the cells to retain in the log phase. There is once or twice exposure of the system into the environment. The key important parameter to be considered in continuous and fed-batch system is the dilution rate. As the medium is fed into the reactor, then the reactor gets diluted and in turn affects the yield of the process. In the simulation studies, the substrate that is being used for given bioprocess, the variable parameters and the quality of output can be simulated in such a way that we can predict the yield of a given process, including the process economics and the efficiency of any given process without using more chemicals and manpower. This simulation can be done in any of the two ways, can be static or the dynamic. The static simulations used in process design simulate the process at steady state conditions, usually at the design operating conditions, and the time is not a variable in the static method, whereas the dynamic models considers time as a variable and simulate the process over a period of time. A dynamic simulation can be used to estimate or illustrate the response over a time to a change in the process. The primary concern of a bioprocess engineer is to develop suitable dynamic models to study the growth pattern and product formation kinetics of any given process. In most practical applications, the environmental or culture conditions can shift dramatically, leading to changes in cellular composition and biosynthetic capabilities. These cellular changes are not instantaneous but occur over an observable period of time. Models can be built on the premise that the dynamic response of the cell is dominated by an internal process with a time delay on the order of response time under observation, where the biomass concentration at any point of time will be determined in context with the yield coefficient, residual substrate concentration, and initial concentration of substrate that was used, where the change in biomass concentration as given by Monod's model is directly proportional to concentration of the biomass and the proportionality constant is specific growth rate of the system, and the equation can be linearized by taking ln on both sides and we get ln xt equal to ln x naught plus mu t, where the xt is the biomass concentration at time t, x naught being the initial biomass concentration, mu the specific growth rate at the time t, wherein in the dynamic model the change in biomass concentration, change in enthalpy and change in concentration of actually living cells can be estimated. The biomass concentration that is being estimated using the common methods will be estimating both the live cells and the dead cells, but practically the number of live cells present in the system provides the suitable yield. So the bioprocess engineer should be capable of estimating the viable cells at any point of time, ignoring the cell death. So the death of cells in a batch culture has to be estimated in context with the death rate constant, and the number of viable cells present in the system should be continuously monitored. And also the product formation kinetics of any given metabolite should be known, and the process has to be optimized according to the kinetics of product formation by using black box techniques equal to the traditional approach to the control of chemical process, it is possible to transfer the functions that can represent the dynamic response of a culture considering only the live cells, and the rate of combustion of the system has to be continuously estimated in order to determine the viable cells present in the system, and the specific growth rate of the cells and product formation kinetics has to be ascertained. In a continuous state tank reactor, to changes in the morphology of the culture can be studied. The dynamics associated with the changes in the culture composition are of great importance to any bioprocess industry, where dilution rate is the significant parameter that affects the efficiency of a bioprocess and is given by the ratio of feed flow rate to the volume of the vessel. The change in biomass concentration is the difference between growth of cells in the reactor and the output of the system. The continuous system of cultivation has been expected to undergo at the steady state conditions, and as at steady state conditions there won't be any change in biomass concentration, then the specific growth rate will be equal to dilution rate, and it is determined that the dilution rate affects the specific growth rate in the system. As per Monod's model, the specific growth rate of the system is given by the change in biomass concentration in the system, but at steady state conditions as mu is equal to D. Therefore, the dilution rate affects the maximum specific growth rate and the substrate concentration, and at the given dilution has to be considered in order to estimate maximum specific growth rate in the system, and this concentration of substrate at any given time in the continuous system of cultivation is given by saturation constant, dilution rate and mu max of the system, and the biomass concentration at any point of time in a continuous system of cultivation is a factor of the residual substrate concentration and substrate concentration at the given dilution, and accordingly the biomass concentration will be estimated considering the yield coefficient, residual substrate concentration and maximum specific growth rate of the system. As the dilution rate increases, the steady state biomass concentration decreases, reaching the steady state residual substrate concentration to provide maximum biomass concentration and maximize the specific growth rate of the system. It is mandatory to estimate the critical dilution rate in a given system in order to estimate the maximum specific growth rate and residual substrate concentration in the system. The fed-batch system of cultivation can be operated either in variable volume fed-batch reactor or fixed volume fed-batch reactor, and the growth kinetics gets varied in both the modes. In the variable volume fed-batch reactor, the volume of the reactor will be varying as the substrate will be fed into the reactor, and accordingly the dilution rate in turn affects and the feed flow rate into the system and residual substrate concentration are the major factors that affect this biomass concentration and yield coefficient in the system. Next, the fed-batch system of cultivation can be operated in fixed volume fed-batch reactor. The fixed volume fed-batch reactors are operated by maintaining constant volume of the reactor. In a fixed volume fed-batch reactor, the limiting substrate is fed without diluting the culture, where the substrate is fed as a very concentrated liquid and feed is provided at a constant rate. The change in biomass concentration is given by G into Y, where G is the substrate feeding rate and Y being the yield factor, and the specific growth rate is given by Gy by X, and the change in biomass concentration is a function of initial substrate concentration and the concentration of biomass at the onset of fed batch and substrate feeding rate, yield factor and the time period. Hence, dynamic modeling is carried out to extract physiological information from any given system to study response of reactor to sudden changes in environment and to study changes in culture composition in due course of time. A simple unstructured kinetic model has been developed to study the dynamic response. The impact of growth period was estimated. The results of experiments optimized through simulation studies can be extrapolated, and it is found that the line exactly fits with the measurement data and simulated data exactly fits with respect to biomass, total cell concentration, product concentration, and also the rate of product formation. And the simulation studies can also be carried out in order to estimate the rate constants and the yield factor in various bioprocesses. The reference books for today's session are Biochemical Engineering by Blanch and Douglas, Biochemical Engineering Fundamentals by Bailey and Knowles, and papers published at PubMed and Science Direct. Thank you everyone for joining the session.